Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sano Genetics. Sano Genetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Andres Moreno-Estrada, who is a trailblazer in population genomics and one of the most influential researchers working to advance our understanding of population genomics in Latin America in particular. He's a principal investigator at the Human Evolutionary Genomics Lab at LANGEBIO in Mexico. And Andres began his career actually working in this field at Stanford University, where he laid the groundwork for some of the work that he's doing today. And my understanding is you worked in Carlos Bustamante's lab for a while, who's also one of the pioneers in this field. We're going to talk about some of Andres's past work, including the Mexican Biobank project, which was on the cover of Nature Genetics and is one of the first and largest biobanks of its kind. And we're going to talk a little bit about what his perspectives are on where we go in the future with population genomics in other parts of the world that are often underrepresented in research historically. So Andres, thank you for joining us and looking forward to the discussion.
Such a pleasure. Thank you for having me.
So to start off here, what initially drew you to population genomics and what got you interested in this field?
Well, to be honest, I didn't know that this field existed. Back in the days, I went to med school actually wanting to understand more about human biology, why we are the way we are, why we get sick, why others do not, where we come from, more like it was really evolutionary questions that you typically do not find answers in med school. However, I knew that med school was kind of a let's think about a tree, and this was kind of the stem of it, to start understanding humankind or human species. Because then the alternative was to study biology, but typically in biology you do not include the study of humans. It's kind of weird enough that in biology, academically, they don't think about humans as animals, right? You only study plants and then non-human animals. So it would have been great to study biology, but I really was convinced that I wanted to learn more about our own species. And, genetics was the door that opened, many keys to answer many cool questions.
So in 2023, you, as I mentioned in the intro, published some pretty groundbreaking work that was on the cover of Nature Genetics. I'd love if you could just walk the audience a little bit through the origins of that work and a little bit about the design of the biobank and some of the major findings from the work to bring people up to speed if they haven't read it before.
Absolutely. If I may, would like to talk about what motivated the biobank, right? Because I had the privilege to lead a project that was before the Mexican Biobank. I think it was in 2014 where we published in Science this first characterization, the first genome-wide characterization of some indigenous groups across Mexico. And that led us to really think, okay, this is a very important first study to understand the structure, how genetic profiling is across Mexico. But it was far from complete in the sense of, it was limited sampling, 15 groups do not represent for the whole diversity of Mexico. And we did find some correlation between ancestry and some phenotypic traits that were important for medicine. In that case was particularly, FEV1, which is, the volume of air you exhale. And, this is in 1 second, which is a measure for diagnosing, pulmonary function, lung function. So I was wondering, okay, what if we had a larger collection that was nationwide and with more phenotypes, we can really have a more comprehensive picture of what the genetic profiling of people and their relationship with disease. And that's how when I came back to Mexico to start my own lab, I immediately contacted people from the National Institute of Public Health. And I was very fortunate to meet Dr. Lourdes Garcia, who was actually looking for someone who could do the genetic characterization of a biobank that they already had collected, like literally 20 years ago. This is the 2020 collection of a National Health Survey that institute already performs periodically. So these are large collection of samples. They deploy a large group of people that make surveys, take measures, take samples. And the original purpose of those surveys is to take a snapshot of the health status of the population together with some biological samples. So they have been doing, for example, measurements of serology, antibodies to see what's the prevalence of responses of diseases, of infectious diseases. But no one had at that time proposed a genomic characterization of such a biobank, which is tremendously valuable. You already have the other phenotypes, and that's what we proposed together. My institution, which is the Senior Staff Research Center, and the National Institute of Public Health, we pitched this idea to a joint call between the UK, the formerly called RC UK, the Research Council in the UK, and the equivalent of the Research Council in Mexico. So this was funded, and this was in 2017, I guess, when we started doing some genotyping. One important point was this agreement allowed my lab to genomically characterize this larger biobank, and we did everything in-house. It was the very first time we embarked in such a big effort to do genotyping. I was at that time also the head of the facility, of the core facility here in Simvestab, that was committed to do genomic services, nationally and internationally. So another good point was that when I was with Carlos Bustamante, we were part of the design of a particular microarray for Illumina, which is a multi-ethnic global array. So we had a particular, access to a discounted price for that particular product because we helped them design it. So that coupled with the fact that we had the biobank in-house, we had a good pricing on that microarray, we were able to have really a rare situation where we could afford doing thousands of individuals genotyping locally for competitive pricing, and everything was done locally. We were able to genotype more than 6,000 samples, out of which only 20 failed genotyping. I think that's a success technically in the sense of, local hands. They did all the, hybridization, sample aliquoting, and I think it's a very low error rate for that. And then also all the genomic analysis and the bioinformatics was done locally. So I was very happy with the success of that particular example, and allowed us to basically have a much richer picture of what the genetic profiling in Mexico looks like.
Yeah, amazing. What were some of the things you were expecting to find, and what did you actually find when you did the analysis?
Right. So we had already, like I said, an idea from the previous study in 2014, but the important the big difference of this newer and bigger sampling is that we cover both like rural and urban areas, right? Yeah. So really what we learn is that there's a continuum of the genetic profile, regardless of, being in urban or rural areas, which means that we have retained a lot of the genetic roots, right, from ancestors that typically are only, regarded as indigenous, isolated groups that probably didn't contribute to, the general population. And actually they do, and by a large fraction. Of course, this varies between individuals. There were, of course, individuals that have very little indigenous ancestry. The opposite you can find in southern states in Mexico, like Oaxaca, Chiapas, that are known to have a large proportion of indigenous population. But is really undistinguishable if you look biologically or genetically an individual in central Oaxaca, the capital city, or in rural areas in the mountains. Biologically, you cannot distinguish which one is an indigenous or which one is not, which tells us also that has a social meaning, that this distinction, right, between these two constructs are merely based on social criteria, not biological, right? So that is important to, give the idea of richness in terms of diversity, and that we share this common root in terms of, admixture between indigenous roots, of course, European ancestry that we all share as well in some proportions. Some others have even African roots also in greater proportions in some areas, even Asian ancestry as well. So we really had a much finer picture of where we can find these four roots, and not just typically think about these two poles of indigenous European ancestry. Is much more richer than that.
Were you able to find any evidence of genetic variants that were either prevalent at a higher rate, that the medically relevant ones that were prevalent at a higher rate in the population, or different genetic ancestry to find subpopulations, or on the flip side, were there others that were less prevalent than other parts of the world, Europe in particular?
Right. So the second, big implication that the biobank had was to, build a database that you can start linking phenotypes medically relevant with the genetic profiling, right? And of course, that was one of the major interests of that paper that was published in 2023. One thing that we were able to model, and this was the work of Dr. Mashal Sohail, who's first author of the paper. She was a postdoc in my lab. Also proudly, I can say that now she's an assistant professor at a independent position. She stayed in Mexico, so also the project helped build local capacity in terms of having new generation of scientists staying local and then training more people, right? So that was great. So what she did is to model various genetic and non-genetic factors that can explain the variation of several phenotypes. For example, cholesterol levels, triglycerides, glucose, and so on, because we know that they are like correlated with a lot of diseases that are of high prevalence in Mexico, like obesity, diabetes, and so on. So surprisingly, one thing that we thought we were going to find related with ancestry was, for example, BMI. That BMI has been typically associated with the fact that obesity is of higher prevalence in Mexico. We have a lot of indigenous ancestry. Maybe that indigenous ancestry is predisposing people to have, problems of obesity, right? What we found was the opposite correlation. So the more the more indigenous ancestry people had in the biobank, lower BMI we were observing in those individuals. So if there's any correlation between ancestry and BMI, it's actually the opposite of what people have been claiming of being, having a higher risk because of genetics, which means that most likely, obesity, it's true that it's a problem. We're not denying that, well, we found that obesity is not a problem. No, there is a lot of people with higher BMI, but genetics is not explaining the higher prevalence of higher BMI. So most likely is due to other factors such as diet, environmental factors that should be taken into account. However, that doesn't mean that we didn't find particular genetic variants in genes that already are known to have an effect in certain direction of predisposing of, for example, higher levels of cholesterol, therefore, cardiovascular problems and metabolic syndrome. So that's true that there are variants, but those are specific variants and not just a general assumption that because you have higher indigenous ancestry, therefore you are at risk. That's what we want to avoid stigmatizing or generalizing, right? So that's the other big thing that we found with the biobank. It's a matter of carefully looking at particular variants and the origin of those variants to know what is the real prevalence. And if you want, I stop there because that's a whole chapter that I would like to talk about what we are working right now in terms of a follow-up of the biobank.
Yeah, I would love to ask a couple follow-up questions, and then let's jump into where it goes next, because that's definitely on my list. I'm also just curious about the way that you were able to draw some interesting insights into some of the population history of Mexico, and I'm really curious to learn a little bit more about what you learned about using this data to go back in time a little bit and reconstruct some of the population history. So maybe you can talk a little bit about that too.
Sure. No, I mean, it's amazing what you can do with bigger sample sizes. For example, reconstruct past migrations or past fluctuations in sample sizes. That's one of the things that we model using the segments of DNA that are identical by design between individuals. This is IBD. And by applying methods with IBD sharing, we were able to, an extension of applications based on IBD is the IBD-NE, NE as a effective population size measure. So what we were able to model is, okay, let's take different areas of Mexico based on their anthropological, zones that the anthropologists have been detecting, but they culturally come from different roots because they are different in South America, Mesoamerica. In turn, Mesoamerica divides itself in several areas. So we grouped our samples based on those anthropological groupings, and then did IBD-NE calculations. And surprisingly, what we found is that patterns, for example, in the Yucatan Peninsula, where Mayan civilizations flourished, and compared to the central part of Mexico or Oaxaca, they changed drastically. If you see the curve, and you can check the figure in the paper, we have a reconstruction of, they start fluctuating basically very coincidentally with the times that we know archaeologically that different civilizations flourish in different places. So Southeast started, first, and then, they collapse when the Aztec Empire then takes over. And then the Aztec Empire goes down right before the colonization period, right? So you can see the fluctuation of different population sizes that coincide with history, which is very beautiful to see that that is captured just with genetics, right? That is agnostic with anything written or told by any story. It's just genetics is telling you how population, behave in the past, right?
Yeah. I find that kind of stuff fascinating. And this is maybe a good segue into what's next. And as you expand the sample size, because there's more to learn on the medically relevant side, but there also we're seeing from these bigger and bigger population genomics programs that you often get concordance between the historical record and what genetics says, but sometimes you get some disagreement as well, where the genetics says maybe the written record on this isn't quite the whole story, or there's more to it or something like that. So maybe you can talk a little bit about what you're working on now and what the future looks like for this program, and anything else that you're working on that's relevant to this.
Sure, sure. I mean, in both fronts, we're actively working in a lot of follow-ups. In the anthropological side, we're interested in looking, beyond what is known already about, Mexico is like a fixed unit of, even within a mixture. And no, there are like distant connections that are relevant to understand what's the makeup of people in Mexico today. For example, there are like accounts that, give evidence of ancestry that you wouldn't expect to find in Mexico, such as Asian ancestry. That's another thing that we found in another publication, right before the Mexican Biobank, but that we are kind of validating with a bigger sampling with the Biobank, is that a particular region in Mexico that's in the Pacific coast of the state of Guerrero, and there's one port, Acapulco, very known for touristic destinations, where we found people that have surprisingly higher proportions of Asian ancestry. So when we saw that, we said, okay, but yeah, it's expected that Mexico has in colonial times known records of people from China and Japan, immigrating into northern Mexico, especially northern Mexico, also in the Yucatan Peninsula, for labor, right? Or economical trade. But then Acapulco was a little bit surprising. And when we looked at the segments, we are able to isolate segments across the chromosome by ancestry, which is what we call local ancestry, and then do a second round of analysis on those segments of Asian ancestry with a reference panel of many Asian potential source populations. So what we found is that they're not Chinese and they're not Japanese. These segments, these Asian haplotypes, are of Indonesian, Filipino, Bornean ancestry, which is, very far away from what we were expecting, right? And then looking at history, it kind of makes sense because there was a galleon, there was a vessel that was making the trip from Manila, from the Philippines, into Mexico, and the port of disembarkation was precisely in Acapulco. And this is known as the Manila Galleon, right? So it was coming from Manila to Acapulco. And as we can see today, that exchange, which was, of course, economical exchange, was not only trading goods, was, of course, also involving, genetic mixing, right? So
yes.
You can see now that had an impact. And there's a lot of also cultural influences of ancient ancestry of Filipino words in Mexico. I mean, everybody knows, maybe if you go to the beach, you want to go and have a beer under a palapa. Palapa is this, wooden structure, to build some shade, and then you can be under that on the beach. That word is of Filipino ancestry, right? So Filipino origin, and many others, right? So there are drinks, there are like some traditional drinks that are particularly coming from Filipino culture as well. So those kind of things, genetics can really be powerful to reconstruct many things that are not necessarily written in history. That particular one had a lot of concordance with history, but there's another one that was even more surprising that we found in Latin America in general. This is not involving Mexico only, but this was a connection between South American indigenous groups and the Polynesian people. So Polynesians also have this long history of navigation. One of the biggest questions in anthropology is whether they made it all the way to the Americas beyond finding new islands across the Pacific, like when they were expanding 2,000 years ago. So we did find evidence that there's some 5-10% of the genetic makeup of Polynesians that has indigenous ancestry. So how can that be? And also taking the haplotypes and isolating those little bits of indigenous ancestry, we are able not only to pinpoint the origin, but the date. So when this haplotype entered into the population in Polynesia. So what we found is absolutely surprising because the date of that exchange was 800 years ago. So that's at least like five centuries before European presence in the region. The region was explored by, the Dutch, French, British, like in the 18th century. But this was found 5 centuries before. So how can that be? I cannot answer that. So I don't know if Polynesians did all the way to the Americas and came back so that we can find Polynesians, individuals with indigenous ancestry, or some indigenous raft maybe ended up, drifted away into the Pacific. I don't know that. But we did find convincing evidence that indigenous groups and Polynesians and indigenous across the globe were moving even before Columbus, right? That we say Columbus was the big navigator and discoverer of new continents. Well, I think he was beaten by other people that not necessarily was written in history, but genetics can reveal it, right?
Yeah, that's amazing. That is really amazing. And it just, it blows my mind to think about that, making that journey at that time, whichever direction it is, or if it was both, to cross the enormous Pacific Ocean 800 years ago. It blows the mind, and evidence. It suggests that people were probably doing it repeatedly, right? It wasn't a one-time fluke journey. It happened many times in history.
We don't know that either. So interesting enough, we found that the signal that I'm telling you, which is some Polynesians having a fraction of their genome of indigenous origin, is not private to one island. So initially, we started a study of genetic diversity in Easter Island, which is Rapa Nui. For many other reasons, anthropologically, it's fascinating, and we wanted also to address a project, focused on their diversity. But we extended the project to other islands, such as the North Marquesas, South Marquesas, many others in remote Polynesia. So we found this signal shared between 5 islands. And then when we compare the specific sequence of DNA, that is the little fraction of indigenous origin among these 5 islands, it is exactly the same sequence. So that could be explained by an event that is of a single time event, single contact. Otherwise, it would be very difficult that you find same segment that could be explained by 5 different journeys, back and forth. Could be, but I think it's more likely there was a one-time event, maybe at the stem of, ramification of Pacific exploration before these islands were colonized. So maybe at some point, before before the founding of these other islands, there was one contact, and it spread the signal in the descendant populations. I think that's more likely, but we don't know for sure.
Fascinating. What do the efforts look like across Latin America more broadly right now? Because there are other major programs in Brazil and other places. I'd love to hear a little bit about how that bigger picture is evolving too, from your perspective.
That's also another very fascinating field because, we suffer a lot from, swimming against the current. That's a good analogy in the sense of, lack of resources. Typically, we're not, powerful nations in terms of getting support from governments. Like, sadly, governments don't have science in their agenda as a priority many times. I think Brazil can be an exception because they really making a bold move, merging efforts between the Ministry of Health and the Ministry of Science, which is something I haven't seen, for example, in Mexico. I would love to, but that's not happening. So we depend really on joining efforts between scientists that are, sharing common goals and common challenges. And we have good examples like the one in Brazil and having, a national initiative of Genomas do Brasil and trying to do genetic profiling across the country. But many others are really lagging behind, right? So one program that I would like to give as an example that we are really successfully joining forces is the Human Cell Atlas initiative, the Latin American branch of Human Cell Atlas. So I don't know if everybody's familiar with this international effort to try to, map the diversity, not at the genetic level, but the gene expression profiling in every tissue of the human body, right? So that's the Human Cell Atlas. One problem that the Human Cell Atlas has, as many other databases in genomics, is the lack of diversity because, tissues and donors that are contributing to the Human Cell Atlas are mostly of European origin. So there was a call, and the Human Cell Atlas has been mostly supported by the CZI initiative, the Chan Zuckerberg initiative. So they launched a call several years ago, specifically to increase diversity representation for the Human Cell Atlas. And we're participating in that call with a whole team of a network of collaborators across Latin America that I'm lucky to coordinate and lead. We are at least 8 different countries, 10 different co-PIs, including Brazil, Uruguay, Chile, Peru, Colombia, Mexico, and so on, and even the US, because we wanted to include also Latinos living in Florida. So with that coalition, we were also fortunate to be funded by CZI, and we are doing, for the very first time, the transcriptomic profiling of indigenous groups across Latin America at the single cell resolution, which is something that goes simultaneously with the challenge of doing this across different countries, but also implementing a new technology that has never been used before, which is single cell sequencing. And we are in year 3 of that effort, and we're getting really good results, really good quality data. Everything processed locally, just like we did with the biobank, with the Mexican Biobank. This is more challenging because we're installing 3 processing hubs, one in Mexico, one in Chile, one in Brazil, so that the nearest location where we collect samples sends their samples to the hub to process locally. And we are near completion in terms of processing data. So it looks great. And we will be, for the first time, doing a genetic map that could be used as a reference for future studies. I want to mention that this is not based on a particular disease, or it's not a case cohort or something. This is healthy population that are donors that are willing to serve as a reference for what is the baseline of genetic profiling in different areas of Latin America. Specifically, I should mention, I didn't say that, specifically in immune cells. So we're starting with 2 tissues because they one of the easiest to collect. One is blood, because we can easily get access to blood and then have this characterization in PBMCs. But also in gallbladder, which is not that easy to get, but gallbladder has an importance from the epidemiological point of view in Chile, particularly because Chile has one of the highest incidence of gallbladder cancer in the world, right, together with India. So we want to have this second tissue included in the Human Cell Atlas to have a baseline also to compare transcriptomics for future disease-based studies. So that's one example of how Latin America is really preparing for having large-scale genomic and transcriptomic or multi-omic projects, locally led, with international funding. And sometimes we need to go out there and because internationally this need is better understood than with our local governments. And I hope this could be a message also to bring the attention of our governments and say, hey, this is important. We need to bet into science, right?
Yeah. What is your sense of what's needed to get the government in Mexico or more broadly across Latin America to invest more in science? Is it about building the next generation of researchers that can drive basic science, or is it more about translational medical research that has more of an impact on the here and now and maybe saves money and saves lives? Or is it a bit of both? I'd love to hear where the political discourse is today and what the big topics are.
Yeah, I think mostly the second, because I think the first is not a problem. Mexico and many other countries have talent, have institutions that are specifically devoted to genomic research. So Mexico has at least 3-4 major institutes that are very focused in genomic medicine or genomic sciences in general. So it cannot be because we lack people or talent, even infrastructure. I would say that is pretty well placed. We just need the fuel to move that train, right? The train is there. We just need the fuel. And the fuel, I think, is we need a political trigger to say, well, this is important, let's do it. And, politics have right now very complicated moments with, other priorities as security. Mexico now is under big fire, to stop drug trafficking. And of course, bigger problem that I think is a missed opportunity not to invest in something that maybe is not seen as a bigger priority right now or a big problem today, but in 5, 10, 20 years, we will miss another train if we're not well prepared with a genomic characterization of our population. Let's think about another pandemic, right? Populations that have already a genomic biobank, such as the UK Biobank, you can, overnight make a GWAS of which is the individuals that carry those variants that are making people having a more severe version of the disease, because they already had the genotypes. You just need to add phenotypes, which is in this case, severe COVID or or mild COVID into that same biobank. You ask people, and you contact and ask people how are they doing. So they were prepared to at least have a much proactive, action against the pandemic. And then in Mexico, we were just fighting in the blind, not even vaccinating with priorities, right? And this is where I see the second point that you made is by giving examples of success, how this can be informative for taking decisions. Hopefully, they can see the priority. I'll give you an example. The Mexican Biobank right now, this is the follow-up study that we're working on right now. First paper was to, give a very fine scale, genetic picture of how the population looks like. But what we want to focus right now is to give access to the population, to the whole database and how they can play with it and really, mine the particular variant or gene of interest that they're working on. So we're building in a platform, and this is the work of one of my PhD students, Carmina Barberena, wonderful student that is building on her own one app that allows people to query what is the frequency of a particular variant that already knows having an effect. This is taking variants from ClinVar, PharmGKB, OMIM, GWAS catalog. And a lot of people is just wondering, what is the frequency of that variant that I know already that has an effect? But in my population, is it relevant? Is it rare or is it common, right?
Yeah.
And at a higher level, for public health purposes, I think it's important for the government to know where is that variant that I know has a very clear risk of higher, or more severe COVID, for example. Where are they? If we had this information in the moment of the pandemic, I could have said, well, let's give priority to a given region in Mexico where to vaccinate first, because where people are going to get sick more severely, right? If you don't have that information, you cannot prioritize resources, right? We have another examples related to, how people metabolize statins. You want to know who is, receiving a particular doses of statins because, again, cholesterol and higher lipid levels is a big problem in Mexico. We have another variant that also dictates or modifies the speed in which people metabolize fentanyl, which is another, highly dependent drug that depends on the dose that you give in surgery, for example, or because you don't want to get more, dependence unnecessarily, you give higher doses, right? So we have now a fine map that tells you where in Mexico there's higher frequency of these variants that are really directly having an effect. And secondly, it's not just a map of frequencies and that's it. Mexico is very complex. The mosaic of diversity is not only how much frequencies in the population, is what is the frequency in the particular haplotype of a given ancestry. And this is where I'm going back to this idea of local ancestry across the biobank, because it's very different if you look at the frequency, the fentanyl variant, for example. If you look at people across the biobank that have European ancestry in that particular position, it's very different, the frequency, if you look at the same biobank, but only those carriers of haplotypes of indigenous ancestry in the same position. That tells you that, there's differential risk depending on ancestry in different parts of Mexico. And that is the next level of care, I think, in personalized medicine that public health should take into account genetic information to move forward. And I think as soon as governments see these as a direct application, hopefully they will start being convinced that more resources are needed to this area.
Yeah, I mean the other example that I'm thinking about that I was curious to ask you about is PNPLA3 in non-alcoholic fatty liver disease, or the disease has been recently renamed to be MASH and MAFLD, but that my understanding is the prevalence in particular people with indigenous ancestry is order of magnitude higher than in some other parts of the world, and it contributes to faster progressing liver disease, for example. This seems like a really interesting and important opportunity to use individual level data to your point, pinpoint is somebody in this high risk group or not, and is there a way to do more frequent screening or some kind of other approach to use this genetic information to have a direct impact from a public health perspective?
Exactly. No, that's exactly one of the things that these kind of databases can help doing, and give more individualized follow-up, like you said, to specific diseases. And this is not only something we are starting to discover from this biobank. This is well known internationally that there's a lot of value of having national biobanks, because we already know that frequencies for those variants or those examples that you're giving are not the same in the UK Biobank or the China Biobank or whatever other in the world, because from evolutionary biology, we understood that we are different. It's not the same that you will have a profile in Africa, in Europe, in Asia, because we have evolved differently and frequencies have changed. So there's a lot of interest internationally that countries, even in Latin America, have their own genetic profiling, right? So Colombia, for example, is building a very similar national database that they want to do through local insurance companies. Colombia is a very particular case that they do promote genetic testing in insurance as as the opposite. You would think that you don't want people, especially insurance companies, to have your genetic information, right? Because could be used against. Colombia is actually promoting that these companies cover the genetic testing because they know they can save even more money by knowing the profiling of the people ahead of time, right? So there's a lot of data already in that regard in Colombia, and Chile has also a national effort to do profiling of the population. So yeah, we know that Latin America in particular has, from the biological point of view, a higher level of differentiation because we come from a much more reduced founding population, which increases drift, and therefore the variants that are maybe in Europe of intermediate frequency, just by random effect through these founder populations, could be just very drastically different, or lower in frequency, or even lost, or higher in frequency due to founder effects. And we have founder effects from both sides. The indigenous founding population and then the European founders were also a subgroup of the European variation. So from both sides, we have a higher, I wouldn't say predisposition, but higher frequency of observing this kind of changing frequencies, right? So yeah, these databases are very important for that.
Yeah, that's a really interesting point. So you have the expansion of the Mexican Biobank program. You have Latin cells work that you're doing. Is there anything else big on the horizon for you that would be interesting for people to know about?
Well, we're also exploring, going back to the anthropological side because it's just fascinating. We're also exploring projects in which we're able to recover ancient DNA from human remains.
I was going to ask about that. Yeah, I'm very curious to hear.
Yeah, no. This is another, big area of opportunity. So it's not that we have a lot of those projects. I mean, I wouldn't consider that our lab is a paleogenomic lab, but we do collaborate with a lot of colleagues that have a lot of expertise with paleogenomics. And once we generate data, is very similar in terms of the pipelines and the methods that we use. So one of the major highlights that we're about to publish, hopefully later this year, is that we found in an archaeological site in central Mexico, we found a very interesting relationship between the human remains we found. We were lucky enough we were able to extract DNA from those, which is something you wouldn't expect also from temperate environments with higher temperatures, and still methods have been improving a lot and more sensitive, so we can use them to extract DNA. And what we found is that there are very different genetic profiles in a single family buried together. So mom and son, for example, and son has a very mixture of signals from northern Mexico and southern Mexico. So this is like an F1 having evidence of, complete different ancestors. You would think that about this isolation in indigenous groups in pre-Hispanic times, but there are some places in Mexico that are actually multicultural, and genetically we're having evidence that actually they were, hubs of mixture or confluence. We don't know, the topological context, and we will tell more in the paper when we publish it. And the other fascinating thing of one of the human remains, the one that is at the very top of the pyramid, obviously, the most prominent individual in that community, was rather carbon dated 1,000 years older than the actual pyramid where it is right now. So that means that we're finding profiling from different regions of Mexico in the genetic DNA and the DNA of one of the remains, but also the dating is having a temporal gap that most likely means that also we have a spatial gap in coming from other place and then founding this new city. So people moving probably with their death, right? Because this was 1,000 years old, older than the actual artifacts and the actual structure, which could be explained by having this big devotion and taking care of the death. As you know,
Wow.
Mexico has a big culture known for,
Yeah.
this Dia de Muertos, which is November 1st, and having this culture of honoring their death. And we are finding evidence of someone that was, taken care of 1,000 years later than
Wow.
the individuals that we found in that pyramid.
That is remarkable. That is really remarkable.
Yeah, yeah, we were very surprised, and that's one example of, yeah, again, giving evidence of the social dynamics that occur in
Yeah.
Mexico in indigenous cultures that pretty much are inherited today somehow, right? So I think that's another tool how genetics can help us to resolve why we are the way we are somehow.
Yeah, I mean, it's amazing on a few different levels. One, that is an incredible anecdote that just underpins the culture that lives on through today, but also that cultural root can run so deep. I didn't know, I didn't know at what point Dia de los Muertos began and how far back it goes, but to your point, it clearly goes back thousands of years in some fundamental way, and that's pretty amazing.
Absolutely,
Yeah.
absolutely.
Well, thank you so much for sharing all this fascinating research with us. I certainly enjoy this, and I learned a ton. I know that people can Google you, find your website if they're interested in learning more. Is there anything that you want to call out that you're looking for, collaborators or funding? If anyone else like Chan Zuckerberg wants to help to support some of these scaling efforts, what sorts of things are most useful for you?
Well, hopefully through these conversations, people get excited about the topic, not only to say, well, that's very cool, but also to find that there are like real opportunities to do great things locally. Let's not take for granted that only great things happen in the powerful nations where typically science is done. I mean, nothing against, science done everywhere else, but typically these things are very not balanced at all. We have big disparities in how science is moving forward, how technology is accessible to different countries. And I think Latin America, as well as other underrepresented areas like Africa, Asia, deserve attention and deserve support to their local researchers so that we can do this with local perspectives. And we have been proven that this is successful in Polynesia, in Latin America. And I think it's really important to motivate local students that are wanting to join this kind of efforts to look at options that we have in-house, right? And we are able to get international level support, funding, and hopefully, speaking of funding, it will come with more.
Yeah, amazing. Well, thank you again for spending the time with us today.
Such a pleasure. Thank you, Patrick.
Thank you. And thanks everybody as always for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sano Genetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.